Let me say I'm delighted to be here in Calgary today and to meet with a number of young people in the faculty, and it's a vibrant faculty, there's no doubt about it. And the people I met with were fun to talk with. Let me say that the Friesen lecture, when I was surprised to be given this prize, and when it happened, I talked to Aubrey Angel and others, and they said, talk about your hepatitis work. I said, I can do that. But I said, I really want to touch a little bit on the COVID issues and the vaccine task force, because so this lecture is really in two parts. The first part is things we've done in viral hepatitis, and the second part is going to be in the things we've done in the vaccine task force. Let me say that it's a tremendous honor to be receiving a Henry Friesen Prize, International Prize. He was the president of the MRC Council from 91 to 99, and he did transition MRC to CIHR. And I had the pleasure of working with him quite closely on some of that, and it was really a terrific time. He also was the founding chair for Genome Canada and really started Genome Canada. And he's won the Canada Gairdner International Award in 1977 for his work on prolactin and the diseases associated with prolactin problems, as well as the the Gairdner Reitman Award in 2001, really around the transition of MRC to CIHR. He is a Fellow of the Royal Society, a Companion of the Order of Canada, and a member of the Canadian Medical Hall of Fame. When I looked in Genome Canada, of course, Alan Bernstein was our first member of CIHR, and Henry went on to head up Genome Canada, so it was quite a formidable pair. And I see previous winners in here like David Naylor and Paul Nurse and a number of others. I really did feel somewhat humbled by this announcement. Let me say that when I came back from Stockholm, I was working on paramyxoviruses and MS. But I did start to see some patients with hepatitis, and particularly hepatitis B. And I think I was the first clinician in Canada in infectious disease to start seeing patients with viral hepatitis, trying to intrude on the gastroenterologist and the hepatologist a little bit. But let me say that hepatitis B was a huge problem. There's about 8 billion people in the world. About 2.4 billion of them have had hepatitis, been infected. Some of them go on to be chronic carriers. And there's still about nearly 300 million chronic carriers in the world, and about 820,000 deaths per year from hepatitis B. So it still is a huge problem worldwide. And I just want to acknowledge Carla Coffin here at the University of Calgary, who's taken on a major role in hepatitis B research in Canada as well. If you look at persistent viral infections worldwide, chronic hepatitis B, as I said, in Canada, there's still quite a number of these as well, about 240,000. Chronic hepatitis C, again, 70 to 100 million, about 1.3% of the world's population. And HIV, you can see the perspective, but they all cause significant number of deaths. And these persistent infections are very important. Let me say that my life changed in reading a paper and preparing to teach students in chronic infection. This was a course in biochemistry at the University of Alberta. In the first year, I was asked to teach about hepatitis B, HIV, and we didn't know about, at that time, hepatitis C. But this paper was published in Cell by Jesse Summers and Bill Mason, and it really showed how the hepatitis B virus replicated. It is the only DNA virus that goes through an RNA intermediate, and it's a very unique mechanism of replication. I won't go into the details, but to just say that this virus enters a liver cell, it gets into the nucleus, and in the nucleus it creates what we call covalently closed circular DNA, which is like a little chromosome being added to the hepatitis B virus, to that hepatocyte. And the real difficulty in curing hepatitis B has been the persistence of that CCC DNA as the source of messengers for the virus to replicate. I can tell you that the therapy we worked on and developed was to inhibit the virus replication in the cytoplasm, but it doesn't really make the CCC disappear. So there's still a major challenge out there in hepatitis research, because we've seen hepatitis C be cured, but hepatitis B, persistent treatment. Just want to make that distinction. But there is some interesting analogies between HIV and hepatitis B. And the activities of the polymerase in HIV, reverse transcriptase, RNase H, and DNA polymerase, were all found in the hepatitis B virus as well. Jesse Summer and Bill Mason came up with their model. And there's one difference, is there's a protein priming from the polymerase. It actually primes the DNA synthesis. And the protein priming is unique because it happens in the virus, but it does not happen in your own cells in your body. So this became a target that we could think about as a way to inhibit this virus without causing serious toxicity in the patients. So I switched my research, and I could just say that I switched from working on paramyxoviruses and MS to working on hepatitis B full-time after I started to think about this. And there was no cell culture systems for hepatitis B, so it made working on this virus somewhat difficult. The only animal models were humans and chimpanzees, and the virus, similar to the hepatitis B virus, was discovered in the early 80s in the Peking duck, and that's what Jesse Summers and Bill Mason used to do this. And I phoned Jesse Summers, and I said, Jesse, has anybody used the duck model to look for antivirals? Because, by the way, I would just say that at this time, if you look at the timeline, 1983, 84, 85, the disease that was dominating the world was HIV. And anybody working in virology was looking for antivirals for HIV. And nobody, because most of this disease is in third world countries, there wasn't many people working at all on hepatitis B at that time. So there was a nice period that I could do some stuff in hepatitis B without having to worry from that major competition. And I asked Jesse Summers if anybody talked to him about this, and he said no. I said, are you interested in doing it? He said, not really, I'm a basic scientist. I said, well, I'd like to just set up this duck model and see if we can't find antivirals, because I started following hepatitis B patients and realized there was so little we were doing for them at that time. You'd follow them until they got cirrhosis or they got liver cancer, and that was not a good outcome. I was very fortunate, and I would say throughout this, to have a very good colleague at the University of Alberta, Dr. Maurice Robbins, who was a professor of chemistry, and he's an outstanding nucleoside chemist. In fact, he was the first to synthesize the dideoxypurines, both adenine and guanosine. And in fact, he supplied these to Sanger, and Sanger did the original sequencing of DNA using the enzymatic sequencing. Sanger had called Maurice Robbins to see if he couldn't get the DDA and DDG from him. Let me say, through skipping a lot of stuff, I ended up getting ducks from different places. There's all kinds of stories about it, but I ended up raising the ducks on my parents' farm. I did this without having a lot of research money at this time, and we set up the colony out there. My dad thought it was crazy, but my mother and dad looked after them, and I'd go out on weekends and pick up duck eggs, bring them back, incubate them, and set up this work to be done. And I would just say across the top of this, the darker the dots, the more hepatitis there was in these. By day 20 in the cell cultures, we'd see a lot of hepatitis B. But when we tried dideoxythymidine, there was the third dot on this was no sign of any inhibition. Dideoxycytidine, very little. ERA-A, which is a drug that I had tried in some hepatitis B patients, had some. But DDA and DDG, and particularly DDG, was extremely potent, at least 10,000 times more effective than DDC and DDT. I mentioned that now, and we wondered why was it that DDG was so very effective compared to the others. Well, I mentioned there was a primer protein, and that primer protein is always, in all of the hepatinoviruses, the first nucleotide bound to it is deoxyguanosine. So when you go in with dideoxyguanosine, you completely shut down the DNA synthesis of the virus. You can incorporate dideoxys downstream, but they do get proofread out. And so that the only one that really had the potent activity was dideoxyguanosine. So we, just looking at the mechanism, it was binding to the primer protein. And when it binds to the primer protein, if you don't have a hydroxy in the 3' position, you don't have any chain elongation, and completely shut down the DNA synthesis after one nucleotide incorporation. Making long story short, we looked at ducks that were chronically infected. These are 12 ducks across the top. Group A was treated with a placebo, 4 of them, and for a period of a week, 2 weeks, and 3 weeks. But you can see when we put in DDG or a prodrug of DDG, after a week, we saw a dramatic decrease. 2 weeks, I really thought at that time, maybe we're going to cure chronic hepatitis B. And the other was the DDA doesn't work as well as the DDG. We did publish this just showing that the purine analogs were not effective inhibitors, the pyrimidine, but the purines were very effective. And that has another interesting, so at this point I knew we were going to, on a good track, maybe we've got something to inhibit hepatitis B. And I approached several drug companies. I approached Burroughs Wellcome. I approached Eli Lilly. I approached Glaxo. And I approached one other one, Burroughs. Anyways, Merck. And I got to say that Merck, Morris Hillerman, who was a fellow I knew well from when I was in Stockholm, met him, approached him about it, and as soon as he found out, I told him it was a nucleoside analog that I thought was a strong inhibitor of hepatitis B. He said, I'd never give nucleoside analogs to anybody because I think they're poison. Well, we've seen that they're not poison and they're used extensively in HIV and hepatitis B and others. Glaxo, Lilly said, no, we think that Jay Huffnagel at the NIH had patented DDC as an effective drug for hepatitis B, based on the work that you saw in HIV where DDC had activity, and they didn't want to get into a patent fight, so they moved out. Wellcome said, well, we've got this all set up in-house. However, when I went to a Gordon conference shortly thereafter, ran into Gertrude Elion, who had won the Nobel Prize for development of acyclovir, and she said, we'd love to work with you, but we have nothing set up for hepatitis B. And I said, well, your management told me that you had it set up, so we didn't pursue it with you. But Glaxo did. There's an interesting thing about Glaxo. When we went to them and met them in Research Triangle Park in North Carolina, presented it to about 9 people, and they brought down Charlie Saunders, who was the president at that time. Charlie Saunders came into the room and said, we're going to work with you, but we need to prove that there's a market for a drug for hepatitis C. Because he said in Western Europe and in North America, if there's a big enough market, we'll work with you. He said, we need 6 weeks to make sure that's true. They came back in 2 weeks and said, you're right, there is a market, and we began working with them. In that, we'd screened a lot of compounds, and we asked to screen lamivudine, which had worked against HIV. We wondered if, because of the similarity, would it work in here? And I'll just point out that on compounds number 18, 20, and 22 were different lots of lamivudine showing very potent inhibition. And lamivudine is interesting because it's synthesized in a plus and minus enantiomer, and the plus enantiomer had no antiviral activity, but the minus enantiomer was very active, and a good antiviral for hepatitis B. It was actually synthesized by Bernard Berlot at McGill, and they spun out a company and it was licensed into Glaxo, and we had demonstrated its activity against the virus. We went back to the duck, and just saying that this is just 3 ducks that we looked at with lamivudine, and these are hours along the side, hours along the side, and these 3 ducks that got lamivudine in high doses, you could see in 24 hours the virus was disappearing. So again, we wondered if maybe we got a cure for hepatitis B. Thought this was going to, and the placebo group didn't show any change. We wanted to go back because we're doing all this in duck, and through Glaxo and Ron Keeney, we had access to a primate colony that had been used for the vaccine studies in New Iberia, Louisiana. And these primates were very well treated. They all had Fisher-Price toys. They had nurses. They had, but they were trained to drink Tang. They had to drink a Tang in the morning and drink a Tang in the afternoon. And we simply spiked their Tang with lamivudine. And down there for a while, I got to say that when I went in to do this study, the owner or the CEO of the primate colony was very tough to get in to put anything new in their animals, and he wanted to know all about the toxicity, which we could give. And I always remember his final question: "Has it been in humans before it goes into my chimps?" Which I thought that was an unusual question, but in fact, it had started to go into HIV patients. So we just increased the dose of lamivudine and took all these samples back to the University of Alberta, and were delighted when we got into the lab and saw how little lamivudine could inhibit hepatitis B in these chimps. These were chronic carriers. Mother chimps are infected, will transmit to the infant chimp. These were infant chimps that had high levels of virus. And I did say there was proofreading. And proofreading can happen in hepatitis B because DNA synthesis is reversible in the presence of a byproduct from the synthesis called pyrophosphate. So pyrophosphorylasis could be reversed if there's a high enough concentration. Well, the virus has very small particles called replicating cores in the cytoplasm, and the concentration of pyrophosphate in there during the synthesis is high, and it will reverse the synthesis. However, we did see there was, and for dideoxys, all the dideoxys, it reversed it, with the exception of DDG, which when you get on that first, there's a difference between the protein nucleoside bond compared to the nucleoside-nucleoside bond. So the DDG wouldn't get proofread out, but when lamivudine was incorporated into the DNA, the pyrophosphate orolysis would not proofread it out. So lamivudine became a very potent antiviral agent, and this just shows the others were all labeled and then proofread out with pyrophosphate in the presence, and you can see the 3-TC or lamivudine was not proofread out. This led to very shortly after that, we had a clinical trial, which we led from the University of Alberta, and we went into with 5 different sites and treated patients. 76 patients, I think, was the number originally, built up to that number, and showed that placebo had really no effect. 5 milligrams a day. When you think an aspirin tablet is about 300 milligrams, 5 milligrams is just like 100th of an aspirin, was still having a big effect on the hepatitis B. 20 milligrams, 100 milligrams, and the standard treatment for lamivudine in hepatitis B is 100 milligrams based on this early study showing greater than 99.9% knockout. And there were big clinical trials run in China, and this is just one showing that on placebo, the disease still progressed. This was designed as a 5-year study. It was stopped after 3 years because there was such a significant difference between the patients on lamivudine and the patients on placebo. And the same thing for hepatocellular carcinoma. Those on lamivudine showed a decrease in hepatocellular carcinoma. Another thing lamivudine did was that it opened the option for transplants in hepatitis B patients with end-stage liver disease. Many sites had stopped transplanting those patients because the liver gets reinfected immediately and often has a more aggressive hepatitis. But we got permission from our local ethics to put a patient on lamivudine and treat them for a few days before the transplant had to be done. And this is the first patient in the world, I think, that got a liver transplant with lamivudine pre and post. And that was done in 1994. Patient did very well, and in the transplant follow-up, 9 years later, somebody stopped the lamivudine, and that was in April. In July, he came in fulminant hepatitis. And today, you always keep those patients, if they are transplanted, on an antiviral to suppress the virus. I would say that also that in cirrhosis, we never knew if cirrhosis was easily reversed. However, in hepatitis B, you'll see across the B is cirrhotic biopsy, and 2.5 years later, a patient with no resistance but on lamivudine showed nice reversal of the cirrhosis. And right now, about cirrhotic patients, about 70% of them will show reversal if you can keep them on antivirals rather than doing transplants and seeing the end-stage disease. Let me say that the evolution of antivirals for hepatitis B, we had been treating with pegylated interferon. It's tough, not easy to tolerate, and not all patients, many patients didn't respond. However, lamivudine was the first licensed compound in 1998, and following that, there are several others. Today, the two best are entecavir and tenofovir. And by the way, they're both purine analogs. Going back to the original mechanism, there's still a major one that has no resistance. So these two drugs are what we use today more than we do lamivudine, because lamivudine treatment monotherapy can lead to resistance in quite a number of patients long term. That's our original contribution to hepatitis B, and we were lucky that it was licensed in 1998 and is licensed in about 200 countries at the present time for hepatitis B. But the others are better drugs, and I completely admit that. Hepatitis C, we also had some contributions to this. Began the research in 1998, and there was no cell cultures, infection in chimps and humans, just like B. And our aim was to develop an animal model for Hep C based on what we could do with the animal model for hepatitis B. And as I point out, collaborators and partnerships are key. Norm Kniedemann is the liver surgeon in Edmonton, and his graduate student, David Mercer, were the two key people that we had in this collaboration. David was transplanting skid beige mice with human hepatocytes. Because they have no immune system, the hepatocytes could. You put them into the spleen, they'd move into the liver within about an hour, and they would start to engraft in the liver. But as you see, at 2 weeks, 4 weeks, 6 weeks, this is measuring human albumin in the serum of these mice, and it would disappear. The mouse still didn't allow full engraftment of this. And I was sitting in the committee, and I said, we need to disadvantage the mouse hepatocytes some ways to allow the human hepatocytes transplanted in this mouse to get an advantage and win. And about that time, one of the people in my lab, Carl Fisher, had been at a meeting and saw the bleeding. You always learn things at meetings, and he'd seen that there was a bleeding studies being done by Rinster, and they had put a transgene into mice in the albumin promoter, so it's only expressed in liver, and 4 copies of urokinase behind it. And this would destroy the mouse's this would destroy human hepatocytes. So we put this transgene into mice, into the skid beige mice, and then we started transplanting with human hepatocytes. And this just shows you the red in this is all the human hepatocytes growing in the background of a mouse with urokinase inhibition, giving it the pale color. But I told you before the albumin disappeared. Now we started to see the albumin at 4 weeks, 5 weeks, 6 weeks. You could see it increasing. And what it really means is these animals had engraftment that kept growing, and we were producing mice with little human livers. And we tried to infect them with hepatitis C. And I got to say that the first time we did this, we had not too good a result. We had 22 animals, and one got hepatitis C. We studied why that animal was different, and that animal was homozygous for the transgene, and all the other mice were hemizygous. So we transferred our colony over to producing homozygous mice, and just as hepatitis C levels in these mice, and the levels are not unlike that you see in chronic hepatitis C patients. So we had good levels of hepatitis C in mice with these humanized livers, and we published that in Nature Medicine, and it received a lot of acclaim at the time as another animal model for hepatitis C, the first non-primate animal model. And this was actually the first mouse that we ever had that had hepatitis C, and it's had some collateral benefits if you're like chimpanzees, because about that time the Institute of Medicine did a study and reported in December of 2011 testing antiviral hepatitis C compounds in chimps was not necessary because we had developed this animal model. So there was an embargo. They've stopped using chimpanzees in a lot of research, and part of it is attributed to this. I can't say that we've had from 2003 to 2018, the NIH had a site at the University of Alberta to test compounds for hepatitis C, and we couldn't keep up in the beginning, so we licensed to Phoenix Bio, and between Phoenix Bio and our labs, we did about 160 compounds testing for hepatitis C antivirals. The work we did on hepatitis B was important to China. China has the highest incidence of hepatitis B in the world, I guess, and they had estimated at that time about 100 million people. And cancer of the liver is their major cancer. And I made a trip early on to Calgary to meet Mr. Lee with Husky. He was here in Alberta with Husky Oil. And we drove into Calgary just as his plane, unfortunately, we didn't have cell phones in those days. His plane was headed to Toronto because they were doing a big merger with Husky Oil, and we couldn't meet that time. However, later, when we had this, approached Mr. Lee about an Institute of Virology, and fortunately, he agreed. And he is a major philanthropist with 15 institutes around the world, including the University of Toronto and the University of Alberta. And he was born in Shantou, and when I first went there, Shantou's medical school was in not very good shape. The buildings were old. However, if you watch the Olympics, the bird nest and all of that, he got the same architect to redesign their medical school. And this is the new medical school, and you can see in the very front there's people walking. It's a very large 9-story building built with this architecture that reminds you of a brainstem and spinal cord in some ways. But by the way, all of those students get trained in cataract surgery, and their medical students go out and do cataract surgery shortly after graduating, because he was very concerned with blindness in China as one of his things. But we established the Li Ka-shing Institute of Virology based on a lot of the, and the U of A had recruited a number of people in virology and immunology, and I would just say we had a strong background in that, and established the institute in 2010. And that was the time of CERCs, Canada Excellence Research Chair, and we had a CERC in virology and recruited Michael Houghton as a member of our place. And Michael Houghton became the director of our Applied Virology Institute. He met with Mr. Lee. Mr. Lee had stressed, I want to see research benefit patients. So he really did stress that, and Michael was of the same philosophy and set up the Applied Virology Institute. The Applied Virology Institute tries to take things we discover in the Li Ka-shing or people and then translate them into benefits for patients. Michael's working right now on vaccines for hepatitis C. We've just done a study, doing a study right now on Group A strep for vaccine as well. And the big thing was that in 2020, October the 5th, at 4 in the morning, I clicked into to see if there was any chance Michael might have won the Nobel Prize. By the way, I nominated him 8 years in a row, and along with some others, his co-developers, or in this case, Charlie Rice, Harvey Alter, and Michael shared the Nobel Prize in 2000. By the way, I saw the flash on the news at 4 in the morning on October the 5th that he had won it. I knew that the committee always phones the people to inform them. So I waited 20 minutes, then I phoned Michael. I actually woke him up. And he said, "What's wrong? Are you okay, Lorne?" I said, "Yes, I'm very good." I said, "You know, did the Nobel Committee call you?" He said, "No." I said, "Well, you've just won the Nobel Prize." So I got the chance to inform him of this. But it was really a tremendous boost for us, for Canada, and Michael continues to be. Let me just say, in this hepatitis work, the key to success is you have to be passionate about it. And as I was seeing patients with hepatitis B and hepatitis C, I always felt very passionate to try to do things for these patients. Persistence is critical. You've got to keep persisting, and there's lots of times you could easily stop doing this work when you run into major blocks. And partnerships. You know, Norm Kneedeman, David Mercer, and Morris Robbins, Michael Houghton, it goes on. So I'm going to go to part 2, some of the time I have left here. In part 2, I'm going to just talk about pandemic COVID-19, and my experience working on the vaccine task force primarily. As we know, this pandemic has been devastating. I think it's over 6 million deaths in the world, about 6.8. Probably closer to 15 million if we know, but this is what's documented. First time life expectancy has decreased in the world by 1 to 2 years. The stress on the healthcare systems you hear about all the time, and there were stresses here before, but the COVID just made it worse. The problems in emergency, long-term care, assessing primary care physicians, it's all related in some way. The economy, U.S. economy alone took a hit of someplace between $16 to $20 trillion by estimates in the U.S. The education, the lockdowns have affected kids in education and socializing. Societal, polarized society like we've never seen, and deepened inequities. There's many other examples of the effect of this on. Let me say that this is a slide from 2018, came out of Johns Hopkins, and it said, we're predicting a pandemic, and it's going to be a respiratory-borne disease, and most likely it'll be an RNA virus, and it'll cause future global catastrophic biological events. It'll have human-to-human transmissibility. It'll be non-trivial fatality rate. It'll be no widely available medical countermeasures. Immunologically naive population. The capability of immune system evasion. It'll be spread by respiratory. It'll be transmitted during an incubation period when people are not symptomatic, and some of the people will remain mildly infected and not symptomatic. And RNA viruses were deemed most likely. Most of this, I think, was focused probably on influenza, but as you know, it was a coronavirus. But you couldn't get better predictions of really what happened, and that's in 2018. 2019, the World Health Organization said the 10 biggest threats in the world were listed here. I just want to emphasize that 6 of them are infectious diseases. So there are still major threats in the world. And this third one, the threat of a global influenza pandemic, is still really there. And we're hearing more about H5N1 and the potential of that as a major pandemic. it's hard for the world to imagine we'd go through what we just did. There's been 11 major viral infections in humans since 1918, and 3 or 4 of them of influenza. That's why people thought it. But MERS and SARS-CoV-1 were pretty significant as well. I'll also emphasize that even in 2019, there was great concern about vaccine hesitancy. And I think that that has proven to be very important. When our committee was set up, it was co-chaired by Joanne Langley from Dalhousie and Mark Lindevan from industry. We had we have 12 people originally. 1 person resigned because they felt there was too much industry on this. I think really we need to see cooperation between industry, academia, and government to handle something of this magnitude. We had an excellent secretariat with Roger Scott Douglas. If any of you know him, just ran a wonderful, wonderful team. We had a lot of ex officio members on there, including the Deputy Minister for ICID and the Deputy Minister of Health. And Anita Anand was the Minister of Procurement, and she and her team were there for almost every meeting. And we were advisory to the Minister of Health and the Minister of Science and Economic Development. And our mandate was, can you help us select vaccines for Canada, and they must be safe, effective, and timely as possible. And we were asked to help looking at rebuilding Canada's biomanufacturing and life sciences ecosystem. That was the second add-on to the committee, and we did some of that as well. Let me say that when I first started on this committee, we had 91 Zoom meetings. None of those meetings were less than 3 hours. We met with many different companies. We had really 266 hours of Zoom at that time. We wrote 52 letters of advice by the co-chairs with input from people in the committees and back to the ministers. I think what was different about the vaccine task force, let me say when I first started, I would wake up every night at 2 in the morning because you realize the responsibility you have and you're trying to come up with solutions for Canada's, there's no vaccines, there's no therapy, and you got this pandemic. And I felt very uncomfortable at times saying, how are we going to do this? And finally my wife said to me, you know, if you wake up every night at 2 in the morning, you're not going to be much use on the vaccine task force, so you better learn to sleep. And we did. There was the urgency of the pandemic, but the advice we were giving was acted on quickly. You often sit on committees, you write reports, the committees gather dust. That wasn't the way this committee worked. And I think it should be thought of as a model of how you might address other key problems, the healthcare system in Canada. Advice was acted on quickly. We had weekly feedback on our advice that we were giving. How is it happening? And I got to give a lot of credit to Anita Anand, who's now Minister of Defense. But Anita Anand was outstanding with her team in procurement. I don't think at the time she got that job, she realized what an important job it would be in Canada. But the approaches to control new viral infections was the public health measures, the closure of public events, social distancing, hand washing, testing and tracing, lockdowns in schools and businesses. Let me say that we did have SARS-CoV-1 in 2002, 2003, and these measures controlled the virus from becoming a pandemic, particularly in Toronto and in Hong Kong, where there was a lot of work that was done very well, and fortunately stopped that. At the time we were going through this, 180 antivirals were being studied in many different clinical trials, vaccines, 237 in preclinical or clinical studies. So it was a little bit daunting to look at all of this in the beginning. But we looked at vaccine productions, and there was new platforms and faster development, particularly the mRNA vaccines and the vector-derived adenovirus vaccines. There were self-applicating vaccines, renew, viral-like particles, and possibly DNA vaccines. And we were faced with these to start sorting out which ones are we to select. Let me say that there was remarkable progress in the vaccines. Vaccines that normally would take 5 to 10 years to develop for a new virus was developed in 314 days. From the time they got the sequence of the virus, the mRNA vaccines were approved, first ones in 314 days. That is remarkable, and I won't go through the details of this, but just remarkable that it happened that way. People, when I've made my trips to Ottawa, I've advocated for better salaries for graduate students, postdocs, and there's a lot of research associates. There's a lot of research associates that get into the system, and it's tough. I just want to say that Katrina Kako was a research associate in many different labs, Pennsylvania and other places, minimal salaries. She, in acknowledging her Gairdner Award, thanked the people who fired her several times because terminated her because she wouldn't have got to where she was if they hadn't. These two people, Drew Weissman, in his lab, working on this, and Katrina Kako, developed the basis. And by the way, we think these vaccines are developed quickly and people just trust them. These vaccines were being developed for 20 years. And Katrina Kako made her discovery in 2005, and they will likely get a Nobel Prize in the next year. I think they will. And she discovered how you could make the RNA stable by putting in pseudouridine for uridine, published that in 2005, and that's the basis of most of the basis of the vaccines. There's another very important part is delivering the vaccines. And how are they going to be delivered? And there's a major Canadian component to this with Peter Cullis at UBC and Ian McLaughlin, who worked at Techmira with Peter, but they developed and discovered the ways you could make this, deliver these vaccines. The mRNA vaccines, when we were looking at them and we saw the first results, let me say the vaccine task force thought the best we see in influenza is around 70%. We were talking if we could get a 70% protection with these vaccines, we'd be very happy. And these, as you know, are we've acquired a number of them. There's really two sets, the Pfizer, BioNTech, and the Moderna. I just want to emphasize Canadian contact once again. Derek Rossi was a graduate student at the University of Toronto under Alan Bernstein, did his postdoc in MIT, and Dubar Afian was a postdoc at McGill. And they are the people who really were the founders of Moderna. And from McGill, Malcolm Sonnenberg, probably one of the world's best RNA biochemists, gave a lot of advice to people on how to stabilize these and do different things. So there's Canadian content, and all of the stuff we put into Canada's basic research is so important in building this background. We were so relieved to see the first results that came out. And of course, the disease was so rampant, it was quick to do these phase 3 studies, but 95% by BioNTech Moderna compared to placebo. Great protection. Same thing with Moderna, 94, 95%. And it was just a relief to see, because we had recommended the RNA vaccines, as we thought they were going to be beneficial. They are more beneficial than we even thought. We also looked at the adenovirus-delivered vaccines, and I, in this lecture, like to emphasize Canadians. Dr. Frank Graham was at McMaster University, and he developed the methods for transfecting cells with calcium phosphate. He generated one of the lab cells that many people use in their lab. That's HEC293 cells. And I asked the question, do any of you know why HEC293 cells are called 293 cells? 293 passages till he got the cells he wanted that worked. And these cells are used around the world to produce this vaccines. And he developed some of the viral-based vectors for gene transfer and vaccine development. Another Canadian hero in this work. Let me say that we procured more vaccines and got criticized that we had about 8 vaccines for every Canadian. At the time we were making these recommendations, we didn't know which vaccine was going to work. So we recommended a number of vaccines, and the Canadian government listened to us in this. Oxford, the AstraZeneca vaccine, Novavax, Pfizer Biotech, Moderna, J&J, Sanofi, GSK. I just want to say the vaccines had an effect. And this is an old slide that I put together a while ago, but it just shows you that in the beginning, at the bottom of the slide, when we had 93 million cases, there were 2.1% death rate. And a year later, after people had 2 of the doses of the vaccine, the death rate dropped to 1%. So that really there was a major benefit and decrease in deaths related to the vaccines. We are dealing with the variants of concern. This is a slide from Graham Tipples in the provincial lab here in Alberta, and it just shows you how one strain is replaced by the second strain by the variants of concern, and how rapidly a new variant comes in, and if it can be transmitted a little better, it replaces virtually 100% of the one that was there before, and you go through the various things, and at the end is the Omicron strains. And the Omicron is a little different, and it's generating more variants, and some of those are clearly resistant to antibodies or evade antibodies and the vaccines better. I just want to say that the R value of these various strains keeps increasing, and the R value is how easy they transmit from person to person. The ancestral strain from Wuhan was an R value of 3.2. Down to the Omicron, it's 9.5. It's as transmissible as measles was, which is considered one of the high ones. We hope that people will get the vaccines. This is just showing that in Canada, with our 38 million people, 84% have the first vaccine, 81% got 2 vaccines. It drops down a lot to 50%, and then down to 12%. Still believe those vaccines and the other doses are very important. We've delivered 86 million, and this is a little bit outdated, but we've delivered a lot of vaccines, but still not enough. In Alberta, this is more up to date, just shows you the red is the first dose, blue the second dose, and the orange is the third dose. And we haven't had enough people in the province getting the third dose. There is not only vaccine anti-vaxxers, but there's also an element of vaccine fatigue. People are just tired of all of this and don't want to think about it more. I just want to say that this picture simply shows, as of February 21st, in the world, we've had 797 million people infected. We have 6.8, almost 6.9 million deaths. But the one that really, the little number at the top, 83,792, is the number of new cases per day. On February 21st, there were 83,700 new cases in the world. I also want to show you that we're not over from this COVID as it is. People out, and we're happy to see people out and doing things, but this is a picture of the vaccine and the number of people per 100 in hospital in Canada with COVID at this time. And we see the earlier peaks, but the final here is between 4,000 and 5,000 in hospital all the time. And people working in the hospitals know that there's still a lot of COVID around. People outside are thinking that there's not much and it's over. It's not really over yet. I just show this slide to say people in hospital, and this is based on a number of per 1,000 patients in hospital, COVID still makes up almost 10% of them. And the second curve, purple one, is RSV, came back. And most of this is children, infants, and some older adults. And at the bottom is the influenza. And you can see that COVID is still the main culprit in respiratory infections and hospitalizations. We've had antivirals developed, but they haven't been as good as I thought they should be. And we have 3 licensed ones, or 4 now, I think, but we need oral, low-cost antivirals that can be used early to change these diseases. And I think we're getting the technologies to do this much better than we have. Of course, the entire protein universe is now getting structures through AlphaFold, and people working in these areas give their sequence of their proteins and get a good idea of the structures. Preparing for the future, the United States has set up 9 centers, collaborative centers, to develop better antivirals. And this is tremendous, these AVI centers, because they are going to develop better antivirals. That's part of pandemic preparedness, which we didn't do before. But we know what the viruses are that are going to cause pandemics in the future, and we need to do this with antivirals and vaccines. There are vaccine centers being set up around these. Canada has responded, and we've seen $2.2 million over the next 7 years committed to pandemic preparedness through CBRIF and through the BRIF. These are major to set up hubs. And there was 9 applications for hubs in Canada. We're down to a few, but I think there's going to be a hub on the prairies. The prairie universities will work together on this because there's some great work being done in the prairies. Our vaccine task force made recommendations for some of the money to build up our biomanufacturing capabilities. You can see here out of the 2.2, we've committed 1.2 has been committed by the government. It's important that we do reestablish a biotech industry for this in Canada. It's important for the graduate students and postdocs here. We need receptors in the community where you can be employed. Let me say the funders, now is the time to invest big, prevent the next pandemic. We can't afford another pandemic. Billion-dollar projects, prepare drugs for the next pandemic. And of course, Cumming, who named the school here, made a very major donation of $250 million to Melbourne and the Institute in Melbourne. Pandemic threats are still out there. When we look at COVID-2, it had a mortality rate of, as I've shown you, 1% to 2%. SARS-CoV-1 that was stopped with public health measures had a mortality rate of 9%, and MERS has a mortality rate of 34%. The other one that worries us in here is, of course, H5N1, bird flu. Sporadically it goes to humans. It doesn't spread from human to human, and that's our big concern, that it stays only with people in contact with animals. But when it has been gone from animals to humans, has a mortality rate of over 50%. We can't afford another pandemic. And if this virus becomes easily transmissible, we have major problems. Pandemic preparedness has changed by this pandemic. I think I used to go to what we would talk about pandemic preparedness, and we were often talking about how will the hospital find some extra places to put patients in ICU, etc. It's not that way anymore. We need to have, Canada had a very good early warning surveillance and real-time monitoring. Unfortunately, we shut that down in 2019, just before this happened. It's being rebuilt. But we need worldwide surveillance, early warning, and real-time monitoring. We need to prepare and stockpile candidate vaccines for the possible viruses that would cause this, develop better therapeutics that I've talked about, improve the diagnostics, and strengthen public health, vulnerable populations, and surge capacity in acute care. These are some of the things we need to do. All I can say is that our message is quite clear. We cannot afford another pandemic. If we fail to take this goal seriously, we'll condemn the world to successive catastrophes with new viruses and pandemics. The pandemic preparedness, in my mind, needs the same urgency as climate change. The problem is when the viruses begin to disappear, everybody relaxes, public health funding decreases. We can't afford to let that happen when you see the devastation that happened with this pandemic. It was a privilege to serve on the Vaccine Task Force for Canada, and one that I will always cherish. And I thank you very much for your attention today. Thank you.